which I'll be giving you a short snapshot of the work that I did here today. So today my talk is called TCR Repertoire Profiling Reveals Clonality of Immuno-STAT primed CD8 T cells. And before I begin, I'd just like to say I don't have any financial or non-financial relationships with the products or services described in this presentation. So today I'm going to be telling you a story about my thesis work that was recently published in the Journal of Virology over the summer, and I highly encourage you all to check it out if you're interested in this work. We have a much more in-depth analysis of what I'll be talking about, so I'm just going to give you pretty much a surface level of what we looked at and how we use the technology described to inform our results. So I'd like to begin by telling you a little bit about the rise in popularity of adoptive cell transfer as a form of immunotherapy that uses CD8 T cells. So ACT, adoptive cell transfer, or adoptive cell therapy, is mainly used to treat leukemia right now, but it's rapidly expanding to treat other diseases such as solid tumors, viral-associated tumors, and autoimmune diseases. Indeed, the global T cell therapy market was estimated to be about $2.8 billion in 2022 and is expected to reach $32 billion by 2030. So this is a huge expanding market. So there's a lot of interest in this field and how to make these therapies better. So with the approval of Tecartus, Yescarta, and Kymriah, a lot of different entities have shifted their business models from the development of small molecule and protein-based treatments to adoptive cell therapies. And there are indeed numerous clinical trials currently ongoing that evaluate both CAR T cells and TCR therapies for a variety of targets. As you can see here, CD19 is one of the most common targets that used in a wide variety of leukemias. And for TCR therapies, you may recognize some of these antigens, such as HPV, a viral-associated antigen, MART1, a melanoma-associated antigen, and so on. So with all that in mind, how do clinicians or laboratory researchers actually produce these T cells for adoptive cell transfer? So CD8 T cells are first harvested from the patient's blood through a process called leukapheresis. Then they're genetically modified using lentivirals or retroviral agents to express either a chimeric T cell receptor, a CAR T cell, or a TCR against their antigen of interest. These cells are then expanded ex vivo in a process called T cell expansion or rapid expansion proliferation, and then they're then infused back into the patient to treat their cancer, their viral-associated cancer, or their autoimmune disease. Alternatively to genetic engineering, you can actually generate a pool of polyclonal CTLs through activation by an antigen-presenting cell, such as a dendritic cell. So this method using dendritic cells has a lot of benefits, and generating these polyclonal T cells has a lot of benefits, such as the ability to target intracellular proteins. If you're familiar with CAR T cells, you know that they're required to generate, or they're required to target a surface cell protein. So by using TCRs, you can target intracellular proteins, which is really important, especially for viral-associated cancers and some cancers that don't actually express those viral or those cancer-associated antigens on their surface. Additionally, they have high sensitivity. These internal antigens are very sensitive for cancer sometimes. These neoantigens are very rare, and so the ability to target these enables a very high sensitivity. And you don't actually have to identify a TCR when you're using this. If you can expand a polyclonal system, you don't need to identify the gene of each one, although it can be important to identify those genes as well if you want to convert it over to a genetic engineering method. Polyclonal TCRs are also very important for a broad immunogenic profile. And so if you have a cancer and you have an immunotherapy that only targets one antigen, there's that potential for antigen escape if you're relying on only one TCR to do the job. If you have a wide variety of TCRs that are able to target the antigen, that may be more resistant to immune escape. And finally, no genetic engineering is required, which removes any sort of risks such as malignant transformation of these cells after you infuse them back into the patient. And so this all seems well and good, but a lot of the times this method uses APCs from the patient themselves, such as dendritic cells. And this results in a very high complexity. As I personally know, it's not easy to make dendritic cells in the lab. I spent a lot of time doing it. It's not a fun thing. It's very time consuming. There's a lot of logistical complexities. It can be very costly. And actually, you get a lot of variability in your result, which results in inconsistent cell qualities. As you can see here, there's a lot of different cytokines needed. It's a very long process, can take up to a week, and you actually get a very low yield at the end of the day. So overall, this results in the high cost and variability, which isn't great for adoptive cell therapies, which you want something very consistent. On the other hand, artificial antigen-presenting cells may circumvent these issues. Artificial antigen-presenting cells, also called AAPCs, provide an off-the-shelf system that can be tailored to each patient without the need to generate fresh dendritic cells for each therapy. Additionally, these modular systems enable fine-tuning of the signals given to CD8 T cells, which potentially can create more potent and longer-lasting CTLs. So we love this consistent quality and this modular system. But what exactly are artificial APCs? What do those look like? So artificial APCs can come in many different flavors. First, they can be either polyclonal or antigen-specific. Polyclonal activators use general activation signals, such as CD3, that activates all T cells, both CD4 and CD8 T cells. And this is very commonly used in things like CAR T cells, where you want to generate a wide variety of T cells, so it activates everything. On the other hand, antigen-specific activators use a peptide MHC1 complex, which will only activate T cells that recognize the target that you've loaded onto that MHC, such as a peptide. Additionally, they can be either cellular or acellular. As you can imagine, cellular APCs are derived from cells. So they can use insect cells, mouse fibroblasts, the K562 cell line, as we previously mentioned, and these are genetically engineered to express these activation signals. So cellular and acellular can also be polyclonal and antigen specific. So you kind of pick one of each half. However, they must be removed from the infusion product, or else the cells themselves can create an immunogenic response, which can be harmful to the patient, especially the non-human cells, such as the insect cells and the mouse fibroblasts. On the other hand, we have the acellular artificial APCs, which use some sort of matrix that is bound to the activation signals, such as latex beads, polymers, gold nanoparticles, or pure protein. But again, a majority of these products also need to be removed from the infusion product because they can generate an immune response in themselves. However, I want to focus your attention on one specific artificial antigen-presenting cell called Immuno-STAT, which was the focus of my thesis. Immuno-STAT was developed by the Almo and Goldstein labs at Albert Einstein College of Medicine, where I did my Ph.D. in collaboration with Q Biopharma. Immuno-STAT is a completely acellular, antigen-specific, and entirely protein-based artificial antigen-presenting cell that is potentially infusible to patients, as it does not contain any synthetic matrix or non-human components. Therefore, it's entirely biocompatible. IST is built from two protein chains. It has a light chain and a heavy chain. The light chain contains the beta-2M chain from MHC class 1, bound to the antigen of interest, so that's modifiable. You can essentially put in any antigen that you want. And the heavy chain contains an Fc backbone, similar to an antibody, bound to MHC class 1. And this is then bound to a costimulatory molecule, which can also take many forms, such as anti-CD28, 4-1BB, or even a cytokine such as IL-2. So this product is actually currently in clinical trials, and previous reports from our lab have shown that the IST framework containing an anti-CD28 domain enabled antigen-specific growth of CMV or HIV-reactive memory CD8 T cells. So taking patients that have CMV or have HIV, we were able to expand in vivo these cells in a mouse model that were able to prevent viral reemergence. Additionally, collaborators have reported the expansion of HPV-16 specific CD8 T cells, which is from HPV, the E7 antigen, using an affinity attenuated IL-2 co-modulator. And this is the product that's currently in clinical trials for head and neck squamous cell carcinoma due to HPV. So all of this work that I've described so far has been using pre-existing memory CD8 T cells. So we're relying on the patient's own immune system to initiate that response, and then we amplify it. However, my thesis had a different idea. What if we can specifically use IST to prime naive CD8 T cells? Could we preemptively target conserved antigens or escape mutants? And targeting naive CD8 T cells would actually enable us to generate de novo immune responses. So we don't need to rely on the patient's own immune system at the beginning. We can infuse these patients with reactive TCR cells that can target a viral antigen or a cancer antigen without them ever seeing it before. So this is essentially a potential vaccine platform. So that brings me to the hypothesis of today, where we wondered if Immuno-STAT could provide sufficient TCR engagement and co-stimulatory signals to drive activation and differentiation of naive CD8 T cells into functional effectors. So let's get into some data. But before we begin, I want to talk about the IST construct that we started using. So this used the MART1 antigen, which is, I mentioned previously, a melon A protein. So it's a melanocyte differentiation antigen that's commonly found in both normal melanocytes and melanoma cells. It's a very common antigen that's currently used in adoptive cell transfer. And it's actually important for us here because it has a unique property in that it has a very high precursor frequency. And what that means is in a normal naive component or a naive compartment, there's a lot of cells that are able to recognize MART1. In fact, it's approximately 1 in 7,500 naive cells in any healthy person that are able to recognize this MART1 antigen. So this is really important for us because it kind of hedged things in our favor. If we're able to get expansion here, that was a really good sign because there's a lot of available cells for expansion. So two constructs were made utilizing this antigen. First, used an anti-CD28 scFv, which is the very classic co-stimulatory marker required for naive activation, and one that did not have a co-stimulatory domain. So this was our negative control that we termed modless. So to go over how we actually stimulated these cells, so first I have a graphic depiction of what we did. First, PBMCs from healthy donors are thawed, and then naive CD8 T cells are isolated using immunomagnetic sorting. We then culture these naive cells with either our IST constructs or dendritic cells that we made in the lab as our positive control. We then culture in media with supplemented cytokines for many days and taking multiple time points throughout this time. And then at day 25, we gather our samples and we run a variety of different analyses on them, including phenotypic analysis, intracellular staining for cytokines, and cytotoxicity assays, as well as immunoprofiling. So now we can actually get into some data. So first, I see here we have a FACS plot showing day 0. And so you can see here we stained with tetramers to detect the antigen specificity of these cells. So in this donor, we have 0.16% of the starting population is able to recognize MART1. So that's our baseline that we're starting from, and we're hoping culturing with either dendritic cells or IST can expand this number. And you can see this is exactly what happened. So here we have representative flow plots at each time point after co-culture with either peptide-loaded dendritic cells on the top row, anti-CD28 MART1 IST in the middle row, and the modless MART1 IST on the bottom. And you can see how both dendritic cells and the anti-CD28 IST expanded a significant amount of MART1 specific population, 45% and 47%. So huge expansion. We were really excited to see this. And it also highlights the requirement for anti-CD28 in this expansion in that MART1 IST that was modless doesn't have any expansion at any time point. And this was very consistent with the multiple donors that we tested. So next I wanted to see if these cells were actually functional. Sure, we can expand them, but can they kill their target? So in order to assess the cytotoxicity of these samples, I ran a europium release assay, which treats peptide-loaded T2 target cells with a molecule that fluoresces upon lysis, enabling us to measure killing through measuring levels of fluorescence. So here you can see I have the different samples that I tested. In black, we have untreated samples, so cells that have not seen any sort of antigen-presenting cell. In pink, I have cells that saw dendritic cells, and in green, I have cells that saw the IST. And as you can appreciate, the IST-treated cells have a really high levels of cytotoxicity at almost all the EDT ratios tested. And specifically, they had higher cytotoxicity compared to the dendritic cell samples, which is a really interesting finding. And additionally, it is a very specific killing in that cells that were cultured with T2 cells without the peptide in the open marks didn't have killing. So it's a very specific killing and very potent killing that the cells that were treated with IST showed. So it was really exciting for us to see. Now, before I get into the next data set, I just want to highlight that antigen recognition is not a one-to-one occurrence. One antigen can not only be recognized by one TCR. In fact, one antigen can be recognized by many unique TCRs. And this is a concept called polyclonality, which I'm sure you're all familiar with. And so we wanted to assess if there's a difference in polyclonality between cells stimulated with dendritic cells versus cells stimulated with the IST construct. So I know Dr. Chenchik kind of went over what this process looks like, but just for our purposes, we assessed TCR diversity through the process called RNA-seq profiling. So we used the RNA version of this process. So this technique uses RNA from flow cytometry sorted tetramer positive T cells. So we sorted our activated cells through tetramer. We isolated these cells and we purified the RNA and sent them up to Selecta for analysis. And so the resulting library was then sequenced via next generation sequencing, and the final data package was processed using R to pull out and quantify the alpha and beta TCR chains, as well as their corresponding CDR3 regions. And I do want to note, because this is a bulk RNA method, I couldn't conclusively make claims about the alpha and beta pairs, but it did enable us to look at the diversity as a whole. So first we looked at total occupied repertoire space. And to imagine what this means, imagine 10 cells that each have a different TCR. Each of these clones, which are different colors, occupy equivalent repertoire space of approximately 10%. So this indicates a high diversity. After simulation, you can now see that there's two clones, the green and the yellow, that are occupying 90% of the repertoire space. So overall, a lower number of clones occupying a higher repertoire space indicates less diversity. I hope that makes sense. So here's the first data that we have, which looks at millions of clones simultaneously. So in dark red represents the top 10 most common clones. First, you can appreciate that in dark blue, in the unstimulated samples, so our baseline level of diversity before any stimulation, you have a high occupation of low copy number repertoire cells. So there's a lot of different diversity going on, a lot of different cells, each with a different TCR. Versus after activation, with dendritic cells or IST, you see there's an expansion in these higher clonotype indices. Specifically, the dark red here in IST is taking up more than 25% of the total repertoire space. So that means in these cells, the top 10 clones are taking up a majority of that space. Versus in the dendritic cells, you can see the top 10 is taking up approximately 15%, and then the next 100 is taking up another 50%, et cetera. So overall, IST shows more occupied space in these top 10 clones, indicating that there's lower total diversity in the IST treated samples. So next we wanted to break down what these top 10 clones are actually looking like. So these pie charts represent the top 10 clones broken down into the unique identified CDR3 regions. As you can appreciate, the top 10 clones occupy 16% of the total repertoire space in dendritic cell treated samples, but 83% in IST treated samples. And this was consistent between analysis of the alpha and the beta chains, which was very important because they should be pairing technically, so they should be relatively equivalent. Furthermore, you can see here the most common clone in IST is actually taking up about 25% of that total repertoire space. So during the FACS isolation, you can imagine we isolated 100 million cells. 250,000 of those are actually a single clone that's recognizing the MAR1 antigen, which was really fascinating to find. So lastly, we looked at if there was any overlap in the CDR3 usage between all the significantly upregulated chains. And overlap here implies that dendritic cells and IST are priming the same TCR chain. There's a match there. So we found 2 overlapping chains for the TRA and 1 overlapping chain for the TRB. And although this might be an artifact for low sequencing depth, it may also indicate that dendritic cells and IST are actually expanding non-overlapping clones, which again was a really interesting finding. But altogether, these data show that IST treatment primes a more focused TCR repertoire than dendritic cell treatment. So that's all well and great for MAR1, but the lab that I worked in was actually an HIV lab. So we wanted to apply these findings to another clinically relevant antigen. So we looked at SL9, which is an HIV-derived epitope from the GAG protein. There's reactivity towards SL9 commonly found in chronic HIV infection, but not so much during acute HIV infection. And indeed, there's no previous success in generating responses against this antigen using artificial APC methods. So this represented the gap in the literature that I wanted to focus on during my PhD. One thing that complicates this antigen is it actually has a very low precursor frequency. So if you remember from MAR1, it was about 1 in 7,500. Here it's 1 in 500,000. So clones that are able to recognize this antigen are much rarer. So we were kind of worried that maybe this wouldn't work just because we couldn't find these clones in our samples. However, we actually found they did expand, which was really, really interesting and really exciting for us. So here you can see that SL9-loaded dendritic cells actually didn't expand SL9 at all, versus anti-CD28 SL9 Immuno-STAT got up to 18% of the total culture after 25 days, which was really exciting to see because, again, this hasn't been done before. We were able to expand HIV-reactive clones in an HIV-naive donor. So essentially, we were able to generate this anti-HIV response in somebody that has never seen the HIV protein before. So this essentially was a potential vaccine platform that may provide protection against HIV in people that are healthy, that don't have HIV. But again, we wanted to do a further analysis on these cells. However, I do want to mention, because the dendritic cells didn't expand, we couldn't do the same comparisons. So instead, we just looked at the HIV responsive clones and kind of compared them to the unstimulated samples instead of the comparison of untreated versus dendritic cell treated versus IST treated. So similar to before, we first looked at SL9-loaded T2 cells, looked at their cytotoxicity, and you can see here that there was significant cytotoxicity at multiple E to T ratios, especially compared to the untreated samples. And indeed, it was also significantly killing over the non-peptide loaded samples. So it was not only significant killing over background, but it was significant killing over the untreated cells and T2 cells that weren't loaded with peptide, which was really exciting to see because it showed that these cells were actually able to target SL9 loaded cells. But additionally, that's not really a physiologic system, especially for viral cells. So we wanted to see if SL9 stimulated cells by IST could actually suppress HIV. So here we did an HIV luciferase suppression assay. So what this is the HIV genome for this specific HIV strain includes luciferase, and that when HIV is infecting a cell, it expresses luciferase and you have a high RLU, so high relative light units. And so when you kill those cells, you actually get a decrease. So that, I just wanted to point that out because it's kind of the opposite as the previous system. So here, because the IST treated samples were able to kill HIV infected cells, we have a significant decrease in the relative light units, indicating that these cells were killing HIV infected cells, naturally infected cells, not specifically SL9 loaded cells, which was also really exciting for us to see because it showed that naive cells from a healthy donor that was treated with IST was able to kill HIV infected cells, which bodes really well for potential future work using this in real patients. But again, we wanted to look at the immunoprofiling. So how does the diversity of these cells look like? Interestingly, these cells were much less diverse than MAR1. However, that kind of made sense to us when we talked about precursor frequency. The initial cell samples that we used, so the initial cultures had about, well, they did have exactly 100 million cells in that sample. And for the MAR1 antigen, 1 in 7,500 of 1 million cells is about 60 to 70 cells per well. So you have a lot of potential cells that were able to be activated. Versus the SL9, 1 in 500,000, there's only approximately 2 cells per well that were able to expand. And that's kind of what we saw here. The top 10 sequences for these 2 samples, which we looked at 2 different donors instead of comparing to SL9 or comparing to dendritic cells. So here we have something called donor 55 and donor 86. And in both cases, the unstimulated samples, again, we have a lot of this dark blue, there's a lot of variety, whereas the stimulated samples, almost 100% of the repertoire is taken up by the top 10 clones. And this itself didn't really tell us, well, how many clones are actually in this top 10? So we looked at the pie charts again, and as you can see, there's 1 clone that takes up over 50% of the whole entire repertoire. And then you have 2, 3, 4 other clones that are taking up a smaller amount of the repertoire. But overall, this was able to generate a polyclonal response as well, which was very exciting to see because a lot of SL9-based immunotherapies now uses just 1 TCR clone, and there's a lot of issues with immune escape. So what we thought is if we could generate a polyclonal response, that may actually help prevent immune escape. So in conclusion, IST is a novel artificial antigen presenting cell that enables naive antigen-specific priming for both cancer and viral antigens. The resulting CTLs from these experiments show that IST is able to enable differentiated, polyclonal, polyfunctional, and cytotoxic CD8 T cells and therefore IST is a potential new immunotherapeutic agent that can specifically be a potential vaccine platform, enabling patients to preventively target either cancer antigens or viral antigens before they even see them. And again, I'd just like to highlight the paper that I recently published on this work. It dives a little deeper into what these cells look like. We look at epigenetics, we look at memory marker assessment, we look at cytokine expression, and there's a lot of really interesting findings there that would have loved to highlight here, but just due to time, I had to kind of take the most important facts here to show with you guys today. And I'd just like to acknowledge everybody that helped me with these experiments, and specifically Dr. Pariso at Selecta, who helped me analyze and understand